Right. So good afternoon, everyone. I'm very glad that you could all join us for this informational evening about the COVID vaccine, and we'll talk a little bit more about the virus itself. I'm very pleased to be joined tonight by Dr. Daniel Ashlock from the University of Guelph. I'm Dr. Joseph Alexander Brown from Annapolis University. Dan, do you want to talk a little bit, I guess, about your credentials and why we're
Sure.
coming to you?
I was hired 16 years ago by the University of Guelph, which is in Ontario, Canada, as an endowed chair in bioinformatics. And basically, my skill is to dig biologists out from under giant piles of data. I've worked on several projects involving viruses, which I personally find fascinating. There are a large number of different types of viruses. They act in a large number of different ways, and some of them, as we'll see in this presentation, can actually be used to devise medical treatments. But we're going to focus on positive stranded mRNA viruses, i.e. the COVID virus, in this presentation. I think I just got one more, so I have 321 peer-reviewed scientific publications. In addition to having an endowed chair in bioinformatics, I'm actually the chair of my department as of last summer, and I'm a full professor of mathematics. So that's me.
And I think
Joey.
the last time we had you here was back early during the first lockdown period, and you visited us digitally in order to deal with that. I'm also glad to be joined by Timur, who's going to be acting as our Russian translator if anybody has any questions and they would prefer to ask in Russian.
Thank you.
The format for this will be along the lines of first a little bit of a kind of an introduction of the biology in terms of what's happening with the vaccine and with the COVID virus specifically. And then what we would like to do is open the floor to your questions and concerns over this, because there is now a vaccine mandate for certain sectors of the Russian economy are required to get the vaccine right now. And the overall vaccination rate is very low within Russia. And this is something that the local governments have been putting various restrictions upon and various mandates about. So given that there is this hesitancy with the vaccine, I would really like if we take some time also to address any concerns that there are from the standpoint of the biology and the science behind all of this. So to start that off, Dan, what exactly is an mRNA virus?
So there's something called the central dogma of molecular biology that says the information in your DNA gets unzipped, it gets written into RNA, in particular messenger RNA, which is then translated in a subcellular organelle called a ribosome into protein. And originally we thought this was all that happened, but it's actually almost completely wrong. It turns out that there are all sorts of branches and things go off in funny directions from that very simple DNA turns into RNA, turns into protein. In particular, RNA can go into business for itself, and that's what these RNA viruses are. You have a protein coat surrounding some mRNA, and then the protein coat manages to somehow insert the mRNA into your cell, and the cell goes, mRNA, I'll make proteins from that, because that's what cells do. And then they go ahead and make the pieces of the viral protein coat, and they reproduce the mRNA. So what happens is you'll have one virus invade a cell, it'll inject its mRNA, and usually it dies, and nothing happens. Occasionally, it takes over the cell, and the cell makes hundreds or even thousands of copies of the virus. So on average, once a virus gets into your body, it starts making more copies than it loses until your immune system jumps up and down on it. And this is again, the point of action. Joey, anything else you want me to say about viruses?
Well, I think in this case, what you mentioned is this viral code. How is it able to kind of get into one of our cells? What's the pathway for that?
There's actually several, but for the COVID, it has something called the spike protein. And I don't like that name. They call it the spike protein, but if you look at an electron micrograph, it's sort of an upside down spike. I would have called it the octopus sucker protein, because what it does is it punches through your cell wall, opens up a passage, and then drops the mRNA in. Furthermore, it's shaped so that it exactly matches a receptor on your cell surface that's supposed to do something else. I mean, this is something that the virus has to have, is the ability to bind with a normally used receptor on your cell surface. And in this case, they just blunder together. Then because it's the right shape, it locks onto the receptor, it opens the receptor, and then it drops the mRNA in. And then your body disintegrates the remainder of the viral package using standard cleanup routines for protein found floating around where it's not supposed to be. But by then the mRNA is in your cell and you're in danger.
Yeah, and this is where then we have the talks about this Ace2 receptor, where it is basically a little thing that normally it's actually a blood pressure regulator inside of the cells. And what happens is that type of hormone binds to it and then releases something known as angiotensin, which is, if angio, you can think of like as vessels. So maybe if you've heard of angina, it's a problem with the vessels. And then tensin, well, stretchiness. So vessel stretchiness. So this is what this receptor normally does inside of our body, is act as a receptor for this to create a different pathway, and it controls and regulates your blood pressure. But the virus is basically co-opting with this spike protein and connecting into it, and that's where we end up with this infection. Okay. So what is an antibody in this case?
Okay, an antibody is an immune molecule that marks or signals something should be destroyed. And in fact, so a lot of my research has to do with using electronic versions of the theory of evolution to try and solve problems. And it turns out that your immune system uses the theory of evolution to try and solve problems. In particular, an antibody is a molecule that binds to and marks for death, something like a virus. The problem is that binds to is not a binary quantity. You don't just bind. You may bind well, you may bind poorly, but the immune system has a feedback loop in it, so that initially it just turns out a bunch of molecules, and some of them just by random chance will bind somewhat to some part of the virus. But when they bind to something, they rip a piece off of it and present it, and that gets fed back to the immune system as a signal, and the immune system goes, this is one of the good ones, make more of it. But, and here's where the evolution comes in, the immune system is not good at making accurate copies of things. Instead, it makes inaccurate copies. Now, this turns out to be good because some of the inaccurate copies actually bind to the virus better than the original thing that was being copied. And so over time, you get things that bind better. Since they bind better, they bind more often and harder, and they send back a bigger signal. And this positive feedback loop of the more you bind to the virus, the more copies of you we make, is a simple version of the theory of evolution that evolves highly effective bind to this virus and mark it for destruction proteins.
Yeah, and I think it's important also with the idea that when you're binding to this spike protein, it's also inactivating the virus. Because if you've got all of these various antibodies clinging on to that spike, it can't infect you.
Yeah,
so
it's like putting a bunch of rubber cement on a nozzle. You can't screw the nozzle on anymore.
Right. I've been kind of relating it close to kind of like different USB connectors. So the antibody is a converter that's making it inactive.
That works too.
Yeah. So with this, how is the vaccines being created? What's their actual role within this?
Do you want all of them, or do you want me to start with Sputnik?
I think maybe we should focus on Sputnik just because it's more applicable to our audience tonight.
Okay, so the mRNA vaccines that you may have heard a lot about are brand new, they're cool. Sputnik, on the other hand, is a well-tested technology. It's one we know works, it's worked for years. What they do with Sputnik is they take an infectious virus, they then treat it to make it incapable of actually infecting. You've got a virus that can invade your cell, but it doesn't contain the directions for making more viruses. So it's a completely castrated virus. But since it can inject stuff into your cells, what it does is it injects the plan for a part of the coronavirus spike protein, the mRNA directions for it, so that the cell will make several copies of part of the spike protein. Now, this is really nice because when you do this, that protein is floating around. It's a foreign protein and it stimulates the immune system. The immune system starts building things that bind to it and mark it for disposal, which means you can get the immune system wound up and ready to attack the coronavirus before the coronavirus shows up. Now, they also did some bed hedging with the Sputnik vaccine. They don't use one adenovirus for this, they use two. I think Joey told me just before we started, he looked it up, they used what?
Stereotype 26, which is actually what the same that they use in the Johnson & Johnson Janssen, which uses only 26,
and then
5, which is used in the Conversera, is the vaccine that uses it. I don't know much about Conversera, but that's also using this. So this is a human endovirus, and the reason for that is you've got the adenoids, which is like your tonsils, and that's where they found these viruses, and that's why they call them adenoviruses, because they're usually found in humans there.
And it's important that they are human viruses because that means they can infect human cells. And in this case, they infect the cell by dropping in directions for spike protein, which is then produced and stimulates a big immune reaction.
Yeah, but
using two different ones is a good idea because one of them may not work. Two of them may not work too, but if you have two different ones, your chances of having it work go way up. The Lancet figure was what, 91.6% effective, if I remember right?
Something like that, yes. And they've done also other tests in San Marino.
Okay, and you were saying?
I was just going to say that part of the difference between the two of them in the first and the second shot is why we're also seeing this kind of benefit of the first shot to the second shot.
Okay.
Yeah. Okay. What is basically this three-phase process of approvals? Because I know that there's a lot of questions about the safety as well as the efficacy.
Okay, that one I didn't study up on, but if I remember right, phase 1 is checking to make sure the damn stuff isn't lethal on its own.
Yeah. Yeah.
You test it in cell cultures, you test it in mice, and you make fairly sure that injecting this into a human being won't make them fall over dead or get green tendrils or some other horrible problem. Now, once you think it's safe to inject this stuff into human beings, you don't really know whether it'll help, though because of the way you made the vaccine, it's likely it will help with the virus. So then the second phase of approval is you have a whole bunch of people who are volunteers, take the virus, but not anything like a production run of how many people you'd want to take it if it works. And you check to make sure it's effective against the virus in this relatively small group. And also they provide a second round of, is this stuff lethal? Because testing in mice and cell cultures doesn't catch everything. Occasionally something, bad happens that doesn't show up there. So that's stage 2. Then in stage 3, they simply test it in a much larger number of people. And this is good because sometimes the bad things that happen are rare bad things and you won't spot them unless you test the virus in a whole lot of people. And so that's stage 1, stage 2, stage 3. And after stage 3, if it's both effective and seems to be fairly harmless, they go ahead and authorize it for general distribution. You may have heard among the people that are objecting to the various coronavirus vaccines that they will not take an experimental vaccine. And here's the thing. By the time you finish stage 3 approval, you are not experimental. The experiments are over, they're done, they're finished. And so this experimental is being used incorrectly as a negative term. In fact, the coronavirus vaccines, Sputnik, Mrna, J&J, Pfizer, these are all well-tested vaccines. The other thing is they do cause some problems, but at a sufficiently low rate that taking the vaccine is much safer than not taking the vaccine. Joey?
So in this, why were we able to do it so quickly? There's this worry that, well, this is probably one of the quickest times between first move to end in these phased approvals processes. Why is it so quick this time?
Partly because they went as fast as humanly possible. Partly because, like Sputnik, I said, is a long-standing, well-tested technology, we already knew those adenoviruses could be properly inactivated and were safe. The only question was, is the protein that we're having them make unsafe? And that's not much of a question either, because the protein's a fragment of the virus protein. So unless for some bizarre reason it's toxic, and most proteins aren't, there's no problem, which meant that they knew ahead of time they could use a fairly short time scale. Unless there was unexpected massive toxicity, this was all going to be fine. Similarly, for the mRNA vaccines, the body tolerates mRNA, it generates mRNA, so this isn't a problem. We don't know how to make an mRNA poison other than by carefully coding an mRNA that makes something toxic. Does that answer the question you were asking? Well, okay, and the other thing is, like I said, this was an emergency. They went as fast as they possibly could.
Well, I think maybe the other thing that we could talk about is just the IRB process and how that was kind of sped up. That a lot of the time in drug testing, you're not necessarily taking time to do the actual test. You're waiting on some giant review board somewhere to get around to the big giant stack of paperwork, which is backed up like 3-4 years by the FDA and a few other places.
Yeah, and you're right. The approval process was expedited in the sense that no vacations, no breaks, no waiting, get it done now.
Thrown to the top of the pile. And I think for anyone that's used to, Canadian or Russian bureaucracy, we know that if you really need to get something done, it can be done quite quickly. It's usually just moving it through the stages of the process, but usually getting through those stages takes months and months and months. In this case, those things were thrown aside. That didn't mean that it didn't go through those approvals. It only meant that we moved it to the top of the pile and said, okay, here's a pot of coffee and donuts. Read the bloody thing and give us an answer. So like even there with the endovirus, I mean, they can, if they have the virus sequenced, how quickly can they do a turnover on a potential spike or a potential antibody?
Okay, I've worked with a firm called Integrated DNA Technologies in Coralville, Iowa. And once you send them the DNA codes for the parts of a virus, they can generally have it synthesized in 2 or 3 days.
Yeah, that was basically my understanding is that we were the actual vaccine itself took about 2-3 days to create, and then the rest of that year was making sure it's safe.
Well, and another thing to note about a lot of these vaccines is that even with the mRNA brand new strategy ones, we've actually been working on them for a long time. This is one reason it's good to have lots of science projects going, is that it'll often turn out that you need one of them real bad. And the inactivated virus, well understood technology, the mRNA, fairly new technology, but one that had been under development for 15 years. Just this was the first time it was ever deployed as a vaccine, partly because up until now, we had all the vaccines we needed, we thought. Another advantage of both these strategies is that you can put them into production very quickly, as Joey noted.
Yeah. So with this, why would you say that it's important that people get the vaccine? I mean, you're doing a lot of work on vaccine protocols. Who should we vaccinate? When should we vaccinate? So what are your thoughts on this?
That's actually sort of sad. So my group spent a lot of time working out who to vaccinate first to try and stop the virus as fast as possible. It was government funded work, and then the government never did anything with it, which is sort of sad. But in general, if you have a population, then for any one virus, there's a fraction of the population where if you vaccinate that many or more, the virus can't escape. In general, a virus has a reproductive number. And if that reproductive number is 1, it means roughly for every person that gets over the virus, a new person catches it. And it's an exponential number. So if it's below 1, the virus dies out pretty rapidly. If it's above 1, the viral infections go up and up and up and up. And these famous waves of the COVID epidemic are an exponential climb, and then people start doing lockdowns and panicking and getting people to wear masks, and it gets to a top and then it trails off. and then somebody decides to open everything up again and another spike starts. Now, in Canada, we've had 4 waves, and the 4th wave is a lot shorter than the 2nd or 3rd one. And it's because we're up to about 70% vaccinated, and that's slowing the thing down. And that's actually the reason you need to be well, 2 reasons you need to be vaccinated. One, it enormously improves your prognosis. If you are vaccinated and get the disease, well, you're less likely to get the disease, much less likely. And if you do get it, you're very likely to have a much less serious case of the disease. But the other thing is, if you're vaccinated, it's much harder for you to spread the disease. And this is the key thing, that we don't want the disease getting spread to more people if we can possibly avoid it. Now, the term for enough people are vaccinated that we're fairly safe is herd immunity. You guys may have watched the premier, is that the right term, of Britain, latch onto the idea of herd immunity, say that's what we need, and essentially propose to let 1 million people die quickly to allow Britain to become immune to coronavirus. Fortunately, he got talked out of that. But in general, herd immunity is what is the fraction of vaccinated people that will stop the virus. And for coronavirus, it was about 80%. And oh, well, too bad, that's not a thing anymore. Because for the Delta variant of coronavirus, it's more like 90 to 93%. You need a lot of people vaccinated. And worse, I believe Europe is currently experiencing Delta plus, and especially Eastern Europe, which put, I mean, okay, where you guys are is a ways from Eastern Europe, but it puts Russia into substantial danger of an even higher level of herd immunity being needed. And just the faster you get people vaccinated, the more you head this off. And then there's sort of a secondary reason, which is that the Delta virus or the Delta variant arose in people that got the virus. And the virus is always tinkering with itself. It's making little changes in its genome, and not on purpose, but because viruses, mRNA viruses, don't copy that accurately. But that means the virus is constantly coming up with new variants. Most of them are awful. You change something critical, you die. But occasionally it changes something that actually lets it reproduce faster. And that's how they know the Delta plus variant in Europe is trouble. It's because it's becoming the dominant variant. It's reproducing faster than the other variants that are around. And in general, that's another reason to get vaccinated as soon as possible. You take yourself out of the, hi, I'm a place where a new variant could arise game.
And I think that there's even a few studies that have shown that if you are vaccinated, the very fact that it has to kind of like work harder to infect you and it has less pathways actually makes it less likely to spread and become more lethal, because it has to turn into a variant that has a slower burn.
This also leads to another thing where I would like to, just sort of applaud Russia. One of the things that is giving me absolute collywobbles about this pandemic is that a lot of places like the United States are going, maybe we should give ourselves a booster before we give, India or Africa copies of the vaccine, and leaving large unvaccinated populations in Africa or, India makes its own vaccines, but not fast enough maybe, is itself dangerous for this same reason of new variants arising. And Russia has been very generous with vaccine diplomacy and deploying its vaccine around the world.
Okay, great. So I guess with that hopeful statement at the end of this, let's maybe open the floor to some questions. Timur is here to act as our translator and local guide if anybody would prefer to ask in Russian, and then he can translate even back your response in Russian. And my first question that I have that was put in beforehand is that many different testings are done on, for example, automotive tests require years, avionics maybe requires decades. How long should these tests be for vaccines? And are we doing it in too short of a time frame?
So short answer, no. The thing is that if you're working on a new type of avionics, you have an existing type of avionics that actually meets your needs. You got planes, no problem. With the vaccine, we need the vaccine 2 years ago. This is a freaking emergency. If we don't have the vaccine, then the number of people that die is appalling. And so comparing testing for something that has to be safe and that we already have something for it isn't really a good comparison to testing for something that we need right now. But we actually have done, I mean, Sputnik, I think there are hundreds of millions of tests have been done in the sense that we have a population of people vaccinated and we have reports on them. So in a way, it's different. With avionics, there's a series of tests you have to perform for like reliability in strange circumstances like heat, low pressure, high pressure, extreme cold. That happens a lot when an airplane's up in the air. But for the vaccine, we don't have to riff through a series of conditions like that. Instead, once we're fairly sure it's not toxic and seems to help, we go ahead and give it to an enormous number of people. And so that's like the difference between serial and parallel computing. I mean, you could run the avionics tests in parallel, I suppose, but that would require a lot of expensive testing facilities. For the vaccine, it's entirely practical to run them in parallel. And so there is a lot of testing. Joey, do you think that needs more answer?
Yeah, the only thing that I would add to that is if you look at where is the failure point inside of a vaccine is going to be during the first inoculation. When it gets into your cell, the RNA runs, it produces the spike, it produces this antibody, and does the antibody bind to yourself? Do you create basically an autoimmune disorder, which we'll able to detect probably pretty quickly? Whereas if you're doing, say, like, the shocks on a car, it's going to have to run for 50 years and damage is a failure point that's far in the future. So our failure point is rather upfront on this, would be my only thoughts.
Okay, you also made me think of another salient point, which is this. The human body is incredibly resilient. A car doesn't fix itself, at least not yet. And even when they do, it'll be a long time before they do it well. The human body, on the other hand, tolerates an incredible amount of abuse before it fails or falls apart. And that means that actually, since we're in a much more resilient system, we can rely on the human body to save itself from minor problems with the vaccine. We always have.
Okay, great. I see a hand raised by Igor. I'll just unmute you there if you want to ask it.
Hi, everyone, and thank you for the talk. Yeah, and just, there is, how would you, answer those who, there is a widespread fear that, this vaccine has been only tested for, X months or a year. What about, what are the guarantees that, in 20 years time, I won't have kids or my kids will have DNA mutations, and all that. There's a big school of thought about the conspiracy theories about the lack of data and the lack of basically ways to predict the long-term effects of this vaccine.
Okay, I would say that there's a lot of data on that. We've been using killed virus vaccines. I mean, I think there are hundreds of them. We've been using them for years, and this has never happened before. So that would require that the spike protein have bizarre, unique mutagenic properties that no other vaccine target has ever had before. And yet, the spike protein analog in other viruses, the thing it uses to get into the cell, is the logical target. So in a way, this is, while, yeah, this is new for COVID, this is not new for medical technology at all. And so I'd say the big answer is, well, it's never happened before and it's had a lot of chances to happen. It sounds like some people are watching X-Men too much.
I was going to say the big question that I see in this conspiracy theory is, can this creation of this antibody change your DNA? So is there a way via the pathways that changes the actual DNA? And since it's putting into your body basically this protein and then using your own body's response system to deal with it, unless right now, if I got COVID and then I gain superpowers, then maybe yes, but it's using a system that's already existent in the body that doesn't change our DNA. So there's not a way that it can via this pathway.
Yeah, the DNA is in the nucleus of the cell. It's actually heavily protected, and the ribosomes are outside the nucleus. So the thing making the protein is nowhere near the DNA. That's another good point. There's no point of contact between the mRNA delivered in the Sputnik virus or the Moderna Pfizer viruses and your DNA. Your DNA is all sequestered. It's safe.
Okay. Igor, I hope that answers your question. Timur, you got your hand raised?
Yeah, you've talked about the Delta variants and successors of the Delta variants. From the evolutionary point, do you think this Delta is one of the last variants, and the virus after this variant is likely to die out quickly?
So my answer to that is that, nah, if we stopped vaccinating today, we'd get 100 more variants. We'd probably, and okay, so here's something from biology. The viciousness of a parasite is directly related to how easy it is to find the next host. And this was first studied in fig trees. There are fig wasps that parasitize fig trees. And when fig trees are 1 tree and 100 in the forest, the fig wasps barely do any damage. But if you get a forest that's 20% fig trees, the fig wasps produce larvae that tear the crap out of the fig trees. And so if we vaccinate or lock down or, stop the virus from spreading, then yeah, the Delta may well be the last dangerous variant we get. If we, on the other hand, allow the virus to keep spreading, it's like buying lottery tickets for variants, and we can keep getting more. So from an evolutionary perspective, this simple result, how vicious a parasite evolves to become depends on how easy it is to get to the next host, tells us what's going on. Vaccination slows down or prevents getting to the next host. Locking down slows down or prevents getting to the next host. And this is also why it's such a great idea to have a sewer system. The sewer system isolates the bacteria that you don't want getting into your drinking water and such like. And not only does it mean you get to the bacteria less, it means it's much harder for like dysentery to find a new host. So the actual dysentery organism becomes more nearly harmless. It becomes a less serious disease. Does that roughly answer the question, do you think?
Sure, thank you. And I also have another question, if you don't mind. From the point of stage by stage approvals that we are now seeing, say in population of pregnant or lactating females, or now we have lots of approvals going on, say for senior age kids, say 12 to 17, and then governments are slowly moving to allow the vaccine to be available to the smaller kids. What's the logic behind that?
So kids are different. That's most of the logic behind that. I note the USDA did just approve the mRNA vaccines we've got in the United States for kids age 5 to 11. So that is moving forward. But kids are different. And also kids have less developed immune systems. Or no, that's more explaining why they're different. And they tend to be smaller. So there's the problem of getting the dose right. There's the problem of, will it actually work correctly in kids because their immune systems are different. And so they did kids last. I'm not sure that was the right decision because, so here's something I often say to junior faculty that have just had kids. School, noun, a place where children go to trade diseases. And in fact, there was a study here in Canada not long ago where in order to save the lives of elderly people, it was found that what you needed to do was vaccinate school children, because almost all the elderly people who were getting sick were catching the diseases from their grandchildren. Now, this was very unpopular with parents. There was a lot of screaming and yelling. And so we said, here's what the math says. And they went, you're criminals. And it was all very, dramatic and irrational. But so that's why we're doing the kids last, is just nervousness about kids being delicate. Oh, and yeah, that's the other thing is parents who are not vaccine hesitant on their own behalf are often vaccine hesitant for their children, just because they're terrified something will happen to their children. And their reasonableness just stops functioning when their children are involved. Joey, do you want to add anything?
Well, the only thing that I would add about kind of the lactating mothers and in regards to that is also it doesn't seem like there's any movement through the milk of any problems with the antibodies there. And in fact, mother's milk is a method of transfer of antibodies from mother to child in that early stage. So one of the worries that you could potentially see, and why they would want to kind of study that a bit more, is there a method of transmission of the virus as well, of that into someone that doesn't have the immune system to build up that resistance. And as far as we can tell, the spike proteins created via at least the mRNA version, I looked up that, it doesn't seem to move via the mother's milk. As to whether it happens via the adenovirus version, which is Sputnik, they've already tested that because we've done it so many times that it doesn't do that type of movement. So I would say a lot of this is out of also abundance of caution.
Thank you so much.
We're always more cautious around children.
Yeah, it's very much more ethics than it is science.
Do we have any other questions?
Okay, at which point I'll just chime in with a couple of facts. When you have an mRNA vaccine or the Sputnik vaccine, this slightly longer, but it also delivers mRNA, it's out of your system. So the regular mRNA vaccines, it's gone in a day. For the Sputnik, maybe 3 days. And every active ingredient in the Sputnik is out of your body. It's all downstream stuff of your immune system reacting to it. So that's another reason why it's very unlikely there are long-term effects is because the compounds involved aren't persistent. They don't stay in your body very long. And just in case it matters, I'm fully vaccinated and I haven't grown any green tendrils yet.
Yeah, and I got the Sputnik, and I'm my normal self. So, you know, if there's any problems, it might be hard to figure out because I've already got enough to begin with.
And I even had a booster.
Ah.
So with that, I guess maybe the only thing else I can say is just, you know, one, is it a bioweapon, Dan? Let's do maybe take some of the things that are being said about it and let's knock them down. Is it a bioweapon?
So short answer, no. Longer answer, if it is an accidental byproduct of bioweapons research, that's interesting, but right now there's no evidence of that. I mean, the Chinese are acting guilty, but the Chinese always act guilty. So that doesn't tell you anything. Beyond that, it's a really bad bioweapon. It takes a long time to kill people, and most people it doesn't kill. So, you know, if I were designing a bioweapon, or if I were trying to order a bioweapon and I got COVID-19, I'd want my damn money back. It's not really a good bioweapon. The other thing is we've found close relatives of COVID-19 in bats and in pangolin and a couple of other things. So this is actually something to worry about for the future. But because the human population is growing and spreading out, human beings are living next to wildlife more and more, and living next to wildlife increases the chance of a virus jumping species from animals to humans, which is where we get a lot of cool diseases, the other SARS things, the bird flu and so on. And for that matter, swine flu is the result of Chinese peasants living right next to their pigs and things like that. So that's a very old example that goes back centuries, possibly.
And even there with smallpox, the original smallpox vaccination was actually from finding out that cowpox and that milkmaids living near their cattle were immune to smallpox.
There was a smallpox variant that lived in cows that would let your immune system get wound up to where it could also fight smallpox.
Yeah. I see that we have a hand raised from Dr. Termosov, the rector. So let's just ask to unmute.
Okay, so can you hear me?
Yep.
Yes.
Yeah, so thank you very much. I have a first vaccination of using the COVID-19, which is a kind of killed virus vaccine. And now in 1 month, probably because half a year will gone, I think what kind of booster I need to use. I can have right now couple of options, like I can use Sputnik like, I can use full 2 vaccine Sputnik, or I can use something else. I'm just curious what is the better understanding. I'm trying to read the information about the, I cannot say recommendation, but the thoughts about this, but that's a very big difference between the different people about what kind of second vaccine should be done. So what do you think? What is your general opinion about the, I'm not talking about whether it's necessary or not. Definitely, unfortunately, probably necessary, because also I check the level of antibodies and it's very low. But anyway, what kind of second vaccine can be it and worse to use it?
So my short answer to that is I don't know, because I don't have the public health data. How vaccines interact and which one will help you, given that you've had another one, is something based on physiology, which means it's very complicated. And the fact that you're asking us after trying to research suggests that you ran face first into that complicated. And so if there's data on which ones helped, that answers the question. Without that data, it's hard to have a good answer, except to say that so far in Canada, we've been mixing shots a lot. And it seems that getting something different from what you had the first time helps. Joey, do you have any information?
So my understanding is that the COVID-19 is, again, one of the enterovirus methods. I'd have to look it up more directly, and perhaps I can get in touch with Alexander later.
It's called inactivated vaccine. It's very, very traditional way of creation.
Okay. So this is very close to more of the Oxford method. Okay.
Yeah,
so
it's not the Mrna, it's not the matrix one, it's not the vector one. It's just a very traditional vaccine.
Right. So it's the inactivated. It's the inactivated COVID, basically.
Yeah.
So in that case, pretty much any of the other ones that are not the inactivated COVID would be different. So that is the Sputnik Light, I think only uses stereotype 5. I would have to look that up again. Or one of the 26, shots of the COVID.
The first one. I think that they use the first one, but yeah. I hear that typically the result of it can be gone in a couple of months. It's like 2-3 months, just only one shot of this first vaccine is not enough. And the main reason for second shot to appear, because you have to make it work at least for half a year. So this is general information I hear about, well, maybe. So the question whether it will be enough just having one, or better to have both 2.
So I think it's interesting that you brought up antibody levels, because I think this is another thing that a lot of people are doing is they keep testing the antibody level and they go, my antibody level is high, therefore I should not get a shot. Dan, do you have any thoughts on that?
Well, yeah, I wouldn't worry that much about your antibody level, because there's a whole nother layer of your immune system that remembers things. If it sees a virus it's seen before, the antibody levels jump very rapidly. If you have high antibody levels, you're currently protected, but you currently have one version of the protection against the virus in the memory of your immune system. So even with a high antibody level, if you have a booster available and you take it, you may be giving, your immune system more cards to play next time the game is convened.
Yeah, and I think that this is another bit, is that people that are recovering from COVID itself could have a very high antibody level, but still could not be protected from a lot of the variants. Because what happens is that the antibody tests that we do are very wishy-washy. They're not very particularized. And with all of the variants that are coming around this, even if you have one of these antibodies that is high, it could sneak around it. So antibody levels are a lot
I
less
also can say
diagnostic.
that we have even more complicated picture, because as for me, for example, I measure two kinds of antibodies. For S, it's RBD corona itself, and for N, viruses. So I do not have anything reasonable for S, but I have something reasonable for N, which is not typically present if I will make the vaccination of such vaccine like Sputnik. So it's another very complicated picture.
Mhm.
So I'm not believing that I need carefully take a look for different figures related to these antibodies and so on. But anyway, it's a kind of indicators.
Yeah, I
So-
think the only advice that we could really say with that, given that we're also both not medical doctors, would be get one that is not of the same pathway of generation.
Mm. Yeah, but which one? Which is just a white one or full size one? It's a more complicated question.
It is, and with our apologies, it is not a question we are qualified to answer.
Yeah, definitely so. And no one, unfortunately, in the world are qualified to answer it. We have only some ideas and thoughts in this moment.
No, the people that can answer that are somewhat in the future.
Yeah, I think the other piece of this is what allows you as well to have the QR code inside of Russia that lets you into a restaurant.
Yeah, and due to this, I'm considering the kind of full-size Sputnik. This is the only reasonable things behind it. But
Yeah, because you know that's the other piece of this is governments are putting in these types of restrictions, and luckily, like Timur's office was able to get me my thing, so I'm able to walk into buildings no problem because I have that. As soon as I go back to Canada, I'm not vaccinated according to them, so I got to go through the whole. I'm going to get 2 more shots of whatever, so that way I can enter Canada, no problem. Dan, any thoughts on the WHO and Canada and all of this, and when we're going to like recognize Sputnik as being actually a vaccine?
Actually, there was a period in which Sputnik was recognized, and then apparently they detected some sort of manufacturing flaw in one batch and withdrew the recognition. And I'm really not sure what's going on. It seems to be an effective vaccine, except there was one batch that wasn't. And for some reason, that's cost it its certification. I'm guessing that as soon as they can certify that they are now checking the manufacturing process correctly, they'll get it back. But you know, predicting the behavior of the WHO is another one of these things that is beyond my capabilities.
Yeah, it depends on whether they recognize Taiwan today or not, too, right? Sorry, that's a little bit of an in joke. Anyone else have any questions? I noticed we got Rabab in the room now. Got a few people that joined a bit late. Does anybody have any questions? Okay, in the interim, I'll give you one more, Dan. 5G cellular networks, do they cause COVID?
There is no physiological mechanism known to man that would allow them to do so. And
So
let me
that's a
follow up by
yes.
saying, you're right. And so it's sort of weird to watch a bunch of people talking about in terror about things like Bill Gates putting a microchip into the vaccine so he can track your every move and monitor you. And they're doing all this complaining on electronic devices that contain a microchip that allow people to track and monitor you.
Yeah, if you want to track and monitor me, this is the easiest thing. Just message me on Telegram and ask me where am I.
But yeah, as far as I know, we don't know of any way to do induced physiology using cellular networks. If we did, it would be the basis of huge numbers of treatments. Here, step into this phone booth for 5 minutes. Okay, we got rid of your cold.
Okay, I think Alexander has another question, or he's just raised his hand again.
Oh yeah, I just tried to say that one of the reason why people start talking about the chipping and so on, I think it was a kind of incorrect advertising of the vaccine, which is called like matrix vaccine, which is assembled like a computer and so on. Immediately people do assume that this is a way to put the chip, computer chip inside themselves. So I think it's a result of improper advertising of the technology, matrix technology behind it.
The reason for this, actually, the genesis of the whole Bill Gates' microchipping everyone is that they wanted to use radio frequency identifier chips to tag people that were vaccinated in Africa, so that way you wouldn't have to carry paperwork around with you from the government. There was a white paper on this, and it was never implemented. It never moved past the, hey, wouldn't it be a neat idea if stage, and then it just went out of proportion.
Okay, that can be a lot of different conspiracy theory around, but I think it's one of the most stupidest idea which I hear about.
Yeah.
I mean that that the vaccination resulting the chipping in 5G and so on, especially the Bill Gates have a power on top of Russian Sputnik vaccine. It's another interesting.
I was having real trouble seeing how Russia would let Bill Gates within a country mile of the Sputnik vaccine.
Igor, you got your hand raised.
Yeah, it's just, one thing is also because there are so many falsifications of the, vaccination certificate in Russia in particular. I don't know if you guys have talked about it when I popped out, but, it's just, there are multiple problems here. And one way for the government to ensure that the person with the QR code have had a vaccine is that they can search for the markers of the Sputnik in the blood sample, from what I understand. And
yeah, but going to the restaurant and being told, please wait 5 hours for your blood test to come back is a bit harsh, yes?
No, what I'm getting at is that this, puts, fuel into this fire of those conspiracy theories. Like, so there is a marker from the vaccine in your blood. So we've told you, which has nothing to do with the chip, right? But, it's still somehow, it's a misinterpretation that there is a way to check whether you've been vaccinated. And if there is a way, then, quantum leap, why not put a chip into your body? So there is this, also going on around here.
Joey and I both have the job of trying to educate people. And every time things like this happen, we sort of die a little bit inside because this just rests on a foundation of no education at all.
Yep. And also, another point, there was, the multiple social polls about, just in Russia, for example, about the people's attitude toward buying the QR code. And, 30 to 50% see nothing wrong in it. And 30% reply that they would have done it themselves. So there was only under 40% of the Russian population in general right now who actually believe that this is a crime and it should be punished, and that it's a bad thing. And this is just a polling.
We're having parallel problems here in Canada. In Ontario, initially, we just had a PDF sent to us by the health ministry saying, yes, you have been vaccinated, and you could edit it and change the name on it. And now we've got QR codes on a phone, and we've had them for about 3 weeks. And I believe that the people forging them are already up and running.
I think that this gets back to the idea, if something is made bureaucratically very, very hard, and it's easier to get a forgery, people will do a forgery. This goes back to like Gabe Newell when it came to the whole idea of like video game piracy, that the piracy is easier than the distribution of the game, so therefore people won't pay for the game. If we make a system where it has too many leaps and bounds in it, and I know even from my own experience of going through this, you have to go to the gologi.com, go through all of that, fill out this thing, show them this, bring in your passport. They flip through it 100 billion times. They put it under a scanner. They they drop it from the floor and see if it turns into, an airplane. They do all of these things to make sure that, what you did is correct before you then get issued the QR code. And I'm a generally tech savvy guy, and it's a huge, giant, crazy process to do this. And what I'm seeing when I go out now and do my shopping is there's always someone at the front that the various, guards are saying, you can't come into the building because you don't have your QR code. It's not usually somebody that's, 20 to 30. It's somebody that is of a more pensioners level who hasn't gone through the whole process. Igor, you wanted to respond?
Yeah, so probably this is something that you had to go here as a foreigner, which is a very unfortunate, right? Because, for example, in my case, I just went to the hospital, and they tell you to bring the paperwork like a passport, and they never asked me even. So all I had to do is just go and take a shot. So I believe it's actually very easy, at least for the citizens here.
Okay, so
to get
you
vaccinated.
Got the shot and,
That's actually somewhat comforting.
Yeah, so
you already had that system set up though, because it's via the SNIL.
True. So I mean, what I believe, here we are talking about 99.9% of the people not vaccinated, not because it's hard or that the clinics are remote, but just because of the various conspiracy theories or just general trend and fashion and so on and so forth. But also I wanted to comment on the buying QR codes. I read an interesting study that showed that people who bought QR codes, they start acting differently. They start acting as if they were vaccinated because that's what they believe. And actually, this is much worse because before they were scared, sitting at home, and now they just turn on the God mode and they go and get sick. So actually, this is worse, for the, in terms of the widespread of the disease. So I've never thought about that.
And it's basically typhoid Mary sort of moving around, right?
That's a really interesting psychological effect. Since everybody thinks I'm vaccinated, I feel vaccinated.
So it's like a placebo kind of, in people's brain, and they just start behaving differently, going out more actively and so on and so forth. And it's just, compounds on top of that. And like there is now a certain layer of people, in Russia who have bought QR code, and now, with all the hysteria, they want to get a vaccine, but they cannot because technically they have been vaccinated, and now they have to wait for several months. So it's really stupid and silly, and I guess no sociologists have predicted that would happen, but it's a really fun case study about this.
I'm guessing nobody asked them if it would happen because that's not real startling.
Yeah, it is.
Yeah, I also want to emphasize that the way how it's checked was also very strange. Today I hear the announce from Euronews that Israel start to allow entrance of the people with Sputnik vaccinated, but also they do require first PCR test in entrance. Second, they do require the antibody tests. And also they say that this vaccination will not work for more than 6 months. That's why you have to be vaccinated not later than 6 months in the second shot before. So this is a kind of overkill from my point of view as well.
Yep. I think the truth about all this is like 5 or 10 years in the future, which is not a good place for it to be, but
yeah, this is very much, especially with any sort of international travel, your mileage may vary. As to whether you're biologically safe is a very different question than whether a government recognizes your passport or recognizes what you've done.
Yep. So I note that it is at least 2 past the hour here in Canada.
Okay, so I would like to thank Dan for joining us tonight.
You're welcome.
In regards to this, and it's always nice to be able to invite you to Annapolis and to get your thoughts upon this all. I will be posting the video to to YouTube, and then hopefully maybe onto the Facebook, because everything has to be Facebook official at Annapolis. I'd like to thank Timur for joining us as well to translate and work that. And I'd like to thank all of you for asking your interesting questions and keeping the conversation moving. So with that, thank you all very much and stay safe, wash your hands.
All right.
Thank you. Bye.
Bye.